Hello and welcome to episode 9 of Gardeners of the Galaxy, a podcast for all of the sentient beings in the universe who have a passion for plants. I am Emma the Space Gardener and I will be your host as we explore gardening on Earth and beyond. My guest on this week's show is Dr Javier Medina, a space plant biologist with the Spanish National Research Council. He works with the European Space Agency to send plant experiments into space and collaborated with NASA on the Seedling Growth experiments, which we heard about from Dr John Kiss in episode 5. Hello, Javier. It's great to have you on Gardeners of the Galaxy.
Hello. It's my pleasure.
So we're going to talk today about plants in space, which is what you've been working on for pretty much your entire career. So for you, why do you think it's important that we learn how to grow plants in space?
Well, it is growth of plants in space is one of the objectives of the space biology, space research in general. Why? Because plants are a necessary companion of humans in any enterprise of space exploration. Apart from other objectives, which are more directly related to basic science, so to culture plants in space can give us some interesting answer to in general, to our knowledge on how plants function. But the direct objective, the objective that mobilizes resources and really is in the interest of the society and the humanity in general, is because plants are, I say, a necessary companion of humans in any space exploration initiative that could be undertaken.
Yeah.
Plants can offer many things that solely plants can offer. They are food, and we can eat, humans can eat more things than plants. But plants offer a mixture of very interesting and unique nutrients, which are necessary, mandatory for the human nutrition. But in addition, plants have an interesting feature of exchange of gases, so that they can contribute to purify the air, to increase, of course, the amount of breathable oxygen. They are the only organism producing photosynthesis, so producing really oxygen and removing CO2. So they are very interesting feature for the human life. And last but not least, plant cultures offer a sort of psychological well-being for humans. So we all of us are accustomed to have plants in our houses, in gardens. We go to the park, we go to the country, and in all these occasions we meet plants and we experience some well-being in taking this contact and taking relationship with plants. And astronauts say, in many cases, astronauts say that this feature of plants is the number one for them, because they put the green point in the spaceship, which produces an interesting, a very, very good feeling for them. So for all these reasons, and probably I have left some other interesting too, well, I think we have a list of things that plants can do, and only plants can do, for making possible the space exploration for humans.
Brilliant, thank you. So you worked on the first European space plant biology experiment, and that was in 2003, and it was carried out by the first Spanish astronaut. So can you tell us a bit about that experiment?
Yeah, it was an interesting occasion. First of all, at that time, we were all fascinated by the International Space Station. At that time, the station had only 2 modules, one American and one Russian module. This coincided also with a very tragic and unfortunate experience, which was the tragedy, the accident of the Columbia, with the unfortunate death of astronauts and so on. And this put even in danger many plants for the future of space research and so on. And at that time, we were fortunate that, really Europe took a very interesting initiative, which was, while we think more deeply whether or not we should continue with the construction of the station and how we can manage all these questions, well, we can mobilize the resources of individual European countries, under the coverage of the European Space Agency, and use the Russian capabilities of traveling, of going with a very well experienced vehicle, the Soyuz vehicle.
Yeah.
So we could organize some small missions, 10 days long, of individual European countries, but all of with the coverage of the agencies, not individual isolated initiative, but really European initiatives. And Spain associated to that idea, and was organized the first Spanish Soyuz mission, which was under the name of Cervantes, our glory, internationally acknowledged, and with the participation of Pedro Duque, now the Minister of Science in Spain, in the Spanish government. The mission and the experiment, which was called Root, was a success. Fortunately, we were capable of growing Arabidopsis seedlings in very difficult conditions, but we succeeded. Yeah. And the analysis gave us an interesting result, which was the disruption of the meristematic activity in the root meristem. Well, probably you will ask what is the meristem and what is the meristematic activity. Well, in the root tip and also in the stem tip, throughout all the life of the plant, there are some cells which are called meristematics, which are like the stem cells, are the mother cells which are undifferentiated, and is a special feature of plants, which is extended throughout all the life. They are undifferentiated, but they are capable of being differentiated to any kind of specialized cell in the plant.
Yeah.
So this meristematic tissue is fundamental for development, because it's the source of cells for plant growth and development. So the specialization of this meristematic tissue is to have a continuous cycle of cellular growth and division. And everything should be, in the normal physiology of this tissue, should be very well coordinated between these processes. Well, we discovered that the microgravity environment was capable of disrupting this coordination. Okay. So this means the result, which was shocking.
Yes.
And this could be very important for the development of plants. If we would like to grow plants, this effect would be deleterious for the normal growth of plants, and was an interesting result.
Okay, so I mean, when you got that result, it must have looked as though it would be very difficult to grow plants in space. But your next series of experiments looked at a way that you might be able to correct that problem, didn't you? You worked with John Kiss and his team at NASA on a series of experiments called Seedling Growth. So what did you learn there that makes it easier to grow plants in space?
Well, starting from the results that we got in the Soyuz experiment, our objectives were essentially two. On the one hand, first of all, of course, to confirm the result. Yeah. And for that purpose, we used the so-called ground-based facilities. So devices or facilities in the Earth, present in different laboratories, that could simulate microgravity, and without the need of going again to the space to try to confirm with other more powerful techniques. Yeah. And so we confirmed that the results that we got in the ISS was really true. Yeah. And was confirmed. The second objective was to try to find a method to counteract this anomaly, this unwanted effect. Yeah. And in that sense, the interaction with John Kiss was fantastic. John Kiss is an expert in the effect of light on the plant physiology. In addition, he had an impressive previous experience on the plant space biology. He had discovered some new tropistic responses to the light in the absence of gravity. So he is an expert in phototropism, and he had discovered in the TROPI experiment that in the absence of gravity, the plants developed a new phototropistic responses to the red light that don't exist at the normal gravity. So we were able to combine in our respective interests. We tried to test whether or not light could be this counteracting stimulus that we were looking for to counteract the effect of the absence of gravity. We had studied in previous papers published in ground research that red light was a very appropriate stimulus for our purpose, because red light was sensed by the cell with special receptors called phytochromes. And these phytochromes produce specifically the activation of those processes that were interested in counteracting the damage caused by microgravity, specifically the cell growth and the cell proliferation. And again, fortunately, we get success. The interaction was fantastic. Our respective teams interacted fantastically. I am very, very happy to have worked with such a fantastic team as the Yonkis one, and with John in person, who is a fantastic person.
Yes, and you won an award, and your team and John's team won an award from NASA, didn't you, for your collaboration?
Mm-hmm.
So that's nice.
Yeah. I was happy really in working in this atmosphere of international cooperation that really exemplifies the objective of the ISS as the most important human, peaceful international cooperation enterprise.
It's nice. It's very nice. So your experiments so far have been working with Arabidopsis, which is the model plant that plant biologists use for study. But you're now working to transfer that knowledge onto edible species. So can you tell us which species you're investigating?
Yeah, up to now, of course, the use of Arabidopsis has been mandatory because, space experimentation has the constraints that it is only very few experiments, and there are constraints of space, constraints of resources, and so on. So Arabidopsis was mandatory because the results should be capable of being analyzed by the scientific community, and scientific community understand the genomes of Arabidopsis perfectly. So Arabidopsis is not edible, Arabidopsis is not especially nice or especially beautiful.
It's a weed.
It's a weed, yeah. But Arabidopsis, of course, has the capability of extending the results obtained in this model species to other. Arabidopsis has also an advantage, which is the family to which it belongs. Arabidopsis belongs to the family of Brassicaceae. Brassicaceae or Cruciferae is a family of plants composed by many edible plants, many plants that we normally use, like cabbage, like broccoli, like mustard, for example, radish. There are many vegetables that belongs to the same family of Arabidopsis. We are now working in a project now in some ground-based facilities together with an Italian group, with Professor Giovanna Arone. We are working in Brassica oleracea, which is the cabbage. We have a contract with Arabidopsis, and we are directly comparing the result of the same experiment performed in Arabidopsis and in Brassica. Of course, this cannot be the only example, and in that sense, the results of NASA with the veggie hardware, cultivating lettuce, has been a milestone in this story. They have been capable of using already different crop plants. Of course, for that purpose, the knowledge obtained in Arabidopsis is and will be necessary. So the use of crop plants doesn't mean that we have to abandon the use of Arabidopsis, because we need to know more basic science in order to be immediately applied. But the transition from the model to the crop plant is being done, and in Europe we are beginning with this Brassicae, more closely related phylogenetically as parents, biological parents of Arabidopsis. In America and in NASA, they are jumping a little bit farther to try directly, but everything has advantages and disadvantages. we got probably more information, more scientific information. They get, well, the direct use of the plant culture in the station. They have snacked the lettuce culture in the station, dressed with olive oil.
Yes. Yes, that was a very exciting moment, I think.
Yeah.
So looking forwards then, because you've been talking about a small greenhouse going to the moon very soon when we have crewed missions. Can you tell us about that?
Yeah, this is a real objective of the first mission with the human presence in the moon. As I told just at the beginning of our conversation, plants are the necessary companion. So if the first astronauts putting the foot on the moon have no plants, this means that the load of the vehicle should be increased, because in any case, they have to eat plants, either if they culture in the moon or they carry with them in the spaceship. So the question of the establishment of this greenhouse is necessary. My group is not directly involved in that, although really the efforts towards these objectives are mostly carried out by means of this special hardware for the use of crop plants that is being installed in the ISS. So they are really the model from which the moon greenhouse will be constructed. Thanks to the experience gained with the American hardware, the veggie, vegetable production system, and also the advanced plant habitat. They are really taking special care in the growing conditions in order to use as much as possible the knowledge acquired in the space research, in the basic space research, and the damages and the stresses, the countermeasures imposed by the stresses discovered to occur in plants. And on the other hand, let's say to the gardener's experience. So how to use the best soil. The soil is a problem, really, to use. They are experiments using the regoliths of the moon. And the discoveries now point that this kind of material is possible to be used, but complemented with other nutrients that make easier for plants to grow and to extract the nutrients from this kind of soil. So this experience of the Americans, of NASA in the ISS, with the use of these new facilities for growing crop plants in the station, is probably the most helpful to get the objective of the moon greenhouses. Now we are engaged in a process of reorganizing, designing a facility which is presently in the European module in the Columbus module of the ISS long time ago, which is the biolab. And we are now engaged in the plant science community, European, in trying to redesign this facility for the use of experiments involving the full life cycle of plants, from seed to seed, to grow plants throughout all the life and to produce seeds in space.
Yes.
And also to extending the use of Arabidopsis to this kind of crop plants. And the European one is still on design, but probably will be very useful because it will have some elements for analysis that the American facilities don't have. For example, the use of a centrifuge for reproducing different levels of gravity. This is very important on the one hand for having a 1G control in space to trying to test the effects of space factors other than gravity is very, very, very important. And also to mimic, to reproduce the gravity of Mars or the gravity of the moon in conditions of space. All this knowledge will make possible relatively easily to pass from the greenhouse in the ISS to the greenhouse in the moon.
So if you were going to join a colony on the moon or on Mars and you could only take one plant with you, what would you choose?
I feel like astronaut and I say a flower could be the best.
Yeah.
Could be the best. I don't eat flowers, but the well-being and the happiness and the relax that offers the culture of a flower and the looking a flower, the meditation capabilities that a flower can, and the relax a flower can can give you is incomparable. But the flower is finally the botanical organism that reconciles you with nature, with the world, and with many other things, and also expresses feelings of fraternity, feelings of humanity. It can be shared. It can represent the best of the human feelings. So probably will be the flower. The zinnia that was cultured in the station is a very beautiful plant
Very pretty,
that
yes.
is worth to be in this kind of enterprises.
Good choice. Lovely. Thank you so much for coming on the show today and sharing your work. It's been fascinating.
It's a pleasure. It was a pleasure. This kind of the space research, space adventure, space travel, space exploration, looking to the space, looking to stars, all these kind of things are so deep in our mind that attracts the interest of humanity in these things. I can say that we have to go there.
We do. So, thank you.
Adiós, adiós.
During the interview, when Javier and I are talking about roots, we should have said that it was the first European spaceplant experiment on the International Space Station. It wasn't the first ever European spaceplant experiment. Now that that's cleared up, that's it for this episode. I want to give a big Gardeners of the Galaxy shout out to Caroline, who very kindly supported the show this week. Thank you, Caroline. Caroline used the virtual tip jar on my website, theunconventionalgardener.com, which is where you'll find the show notes for this episode. If you want to become a regular supporter of the show, you can sign up via patreon.com/Gardeners of the Galaxy to gain access to extended episodes and bonus content. I'll be back next week with an interview with Joya Massa, one of the NASA scientists working on the veggie growing system. Joya and I will be talking about the challenges of growing plants in space, how NASA selects the best crop varieties, and when astronauts might be able to tuck into that first space tomato. Thanks for listening. Goodbye.
Orbital Gardens, this is Mission Control. We are confirming termination of your signal. Unfortunately, we have also lost your movie requests, so we have picked you The Martian and Frozen. Uplink commencing now. Mission Control out.